I just want to welcome everybody to the last visiting professor program for this academic year. Really a fantastic lineup we've had, and today is going to be the great culmination. Just to acknowledge the people who put a lot of time and effort into each and every one of these. The fellows who put together the monograph worked very, very hard to put it together and make a wonderful presentation. In September, we had Glenn Levine and Catherine Michaelis, who's on her way to Texas to start her fellowship in heart failure, presented. Bruce Lerman in October. Raman Sharma, who got married around 2 weeks ago, did that, Controversies. Steve Marks was here in November. Luke Murphy did his. In December, Dr. Januzzi from Mass General and Dan Masacanto did a wonderful job with biomarkers and heart failure. Martin Marin was here in January, and David Narotsky worked with him on hypertrophic cardiomyopathy. Harlan Krumholz in February, and Mike Davalis worked with him. In April, Glenn Fishman. Josh Kiss did an excellent job, and we had in May, we had Scott Gottlieb, who was head of the FDA, who had a one-on-one sit-down, and Dr. Fuster did an excellent job with that. And today, no small feat of getting back here Colin Mcrae from Brigham and Women's, and Anna Plitt has really done a wonderful job putting together a very difficult, very broad topic, impact of genetics and big data on cardiovascular disease. I'll just tell you who will be on the panel. And one of the extraordinary things at a place like Sinai is to get from the moderator on up a fantastic panel, which really, in Anna, in her summary, next to last penultimate paragraph, really describes that cardiovascular diseases and diseases in general really need a collaboration of experts in many, many different fields, as she's going to explain in more detail. But our panel really typifies that. Dr. Hajar, Arthur and Janet Ross, Professor of Medicine, Director of Cardiovascular Research Center, is the moderator. The panel includes Johan Bjorkegren, I hope I didn't mispronounce that too badly, Professor of Chief Clinical Science Officer, Department of Genetics and Genomic Sciences, Bruce Darrow, Senior Vice President, Information Technology, Chief Medical Information Officer, Mount Sinai Health System, Bruce Gelb, Gogol Family Chair and Director of the Mindich Child Health and Development Institute, Director of Center for Molecular Cardiology, Professor of Pediatrics and Genetics and Genomic Sciences. I told him that Dr. Mcrae gave him a shout out noontime for helping him with his research. Amy Kontorovic, who is Medical Director of Adult Cardiovascular Genetics, mentored very ably by Dr. Gelb. Jason Kovacek, Principal Investigator, Kovacek Laboratory, Associate Professor in Cardiology. Mark Miller, Cardiac Arrhythmias Service, Professor of Cardiology and Electrophysiology. And without further ado, I've given her a big lead-in, Anna Plitt.
Thank you, Dr. Mcrae, for coming down. Thanks to Dr. Fuster, Dr. Goldman, and all of the panelists for organizing this presentation. So I will give a quick summary on the impact of genomics and big data on cardiovascular disease, and we'll be focusing on atrial fibrillation. So as you all know, afib is the most common cardiac arrhythmia worldwide, and in the US, it's projected to affect over 6 million people by the year 2050. It is associated with significant morbidity and mortality, and despite its prevalence, we are still learning about this disease and how to treat it. For decades, afib was thought to be a result of comorbid conditions, but we have learned that multiple reentrant circuits, fibrillatory conduction, and autonomous foci are involved in triggering and sustaining this arrhythmia. Within minutes of afib onset, there is atrial remodeling. And we are just now beginning to recognize the concept of atrial cardiomyopathy. And researchers are starting to look into genetics for answers. The genetic basis of afib is a relatively new concept, and the first gene was actually identified in 2003. Since then, GWAS studies have identified genes encoding cardiac ion channels, sarcomere proteins, and transcription factors. Understanding the earliest changes in AFib prior to environmental modifiers will give us insight on the potential biologic mechanisms of spontaneous or sustained arrhythmia and help us to better understand the various subtypes of AFib and also how to treat this condition. While arrhythmia detection by an EKG remains a gold standard, a large number of patients with paroxysmal or symptomatic AFib are missed during routine screening. New methods of detecting AFib have been developed relying on already implanted pacemakers or defibrillators, and most recent trend using smartphones. Device-detected subclinical AFib, or SCAF, has been noted in over 40% of patients who have long-term rhythm monitors, and it has been associated with increased risk of thromboembolism. And this raises an important question about treatment. We are definitely seeing a dissociation between the temporal relationship of having the arrhythmia and developing a stroke. And so what does this mean about patients without known documented AFib? Does the duration of SCAF matter? Should we be using anticoagulation in this population of patients? The IMPACT trial was the first randomized trial to use continuous device-detected arrhythmia to guide initiation and withdrawal of therapy. And while it was stopped early for futility, there are 3 other ongoing trials to address this question. As can be seen, there's a lot of interest in understanding AFib and to guide therapy better. And this is where genomic medicine comes in. Genomic medicine aims to improve the prediction, prevention, diagnosis, prognosis, and treatment of cardiovascular diseases by using large-scale data. It aims to incorporate the omic data and the microbiome. The omic data refers to data within the genome, while the microbiome describes the non-human genetic data within the human bodies. And one example of such research is the One Brave Idea designed to address the coronary heart disease led by Dr. Mcrae. The goal of One Brave Idea is to identify earliest subclinical indications of the disease in childhood and use behavioral and pharmacologic interventions to fully eliminate atherosclerosis. It will use passive, non-invasive data collection on a large scale via wearable devices incorporated in individuals' daily life. This will allow for detection of intricate details that are otherwise lost when the switch from healthy to the disease state occurs. Now, could a similar framework be applied to AFib? And I propose that this is already happening. For example, the Cardiaband technology allows the person wearing the watch to record the rhythm strip and transmit the information to an iPhone for analysis. This smartwatch is the first to be cleared by the FDA to be available to the general public without a prescription and speaks to the evolution of mobile healthcare technology to obtain real-time recordings of physiology and to provide diagnostic information. However, we must consider several logistical and ethical issues that arise with the introduction of any new technology. Specifically, the security of the platform that holds this data, accessibility to general public, and legal ramifications for the physicians need to be addressed. And lastly, genomic best practices that deal with informed consent, privacy, and confidentiality will need to be implemented, and patients and providers educated on this evolving field. Thank you.
Thank you very much, Anna, for an excellent job in addressing a very fascinating field evolving so rapidly. And it's a pleasure today to address this field, genetics, in the cardiovascular field, Dr. Callum Mcrae. I would say that his career has 4 different phases. Phase 1, of course, was his birth. He was born in Scotland, and then he actually obtained a degree of medicine at the University of Edinburgh, and then the MRCP from the Royal College of Physicians in the UK. And then he had his PhD in the field of human molecular genetics in London. Now, he was a senior house officer at Hammersmith, and then he obtained his register in medicine at the Royal Infirmary of Edinburgh. In fact, he joined the Royal Infirmary of Edinburgh about a few weeks after I moved from Edinburgh to the Mayo Clinic. He was then, it goes into the phase of research fellow now in the United States. He moved to the United States and he was working with in the Dr. Seidman laboratory at Harvard. And then here he obtained in Boston, he had the internship and fellowship in cardiology. If I am correct, his internship and residency in medicine was at the Brigham, and his fellowship in cardiology was actually at the Mass General. And this goes into phase 3. He decides to move from the United States back to the UK, where he was lecturer at St. Joseph's Hospital. And then he said, you know what, I rather like United States. So he decided to come back. And, you know, he had a number of positions between the Mass General and the Brigham, and it's fascinating. He goes from one institution to the other. And so that's a real asset to move in Boston between two competitive institutions and to go back and forth, back and forth. Anyway, he became the training program director of the Cardiovascular Disease Fellowship at the Mass General, and then finally became clinical director of the Brigham Genomic Medicine and head of cardiology at the Brigham. Now, these different phases have really developed an individual with lots of accomplishments. He has been in terms of his grant review activities, he has occupied key positions at the National Cancer Institute, National Heart, Lung and Blood Institute, National Institute for Neurological Disorders, British Heart Foundation, European Research Council. So important positions in terms of evaluating the science and grants of research. He's an editorial board member, of course, of the Journal of the American College of Cardiology, and then he's in the section editor of the new journal, Basic and Translational Science of the Journal of the American College of Cardiology. He's in the editorial board of Cardiovascular Genetics and Genomics, Circulation, Journal of Molecular Medicine, etc. Now, in terms of honors and prizes, he was finalist of the Samuel Levine Young Clinical Investigator Award of the American Heart Association, the Dunn Award of the Brigham and Women's Hospital, Roman DeSantis Award of the Mass General, and he was a nominee for Excellence in Mentoring Award from the Harvard Medical School, and then at the Brigham he obtained the Joss Thorne Award, a very prestigious one. I think what is perhaps most impressive is his funded projects from the National Institute of Health. In fact, over these years, over these different moving back and forth, he obtained 41 grants that I was able to review today in his CV, and from those, 50% actually are grants from the NIH, actually 20 over a period of few years. So this is no question, is a great accomplishment. Now, the last one, of course, is quite important, and which is the American Heart Association and Google and so forth, which is, you know, I think he got close to $80 million, nothing. You keep them at home? Yeah, it's probably the largest grant I have ever seen, given to a single individual who he's promised to discover the world of things that we don't know yet. So great expectations over the next few years. He's very good clinician. In fact, some of the awards, like the Roman DeSantis Award, was given to being a great clinician. And at the same time, he's a fantastic teacher with lots of commitments in teaching. Actually, it's not unusual to see great investigators who are great teachers and at the same time great clinicians, and he's one of them. Well, I finish by saying that his curriculum vitae has obviously the most important aspect, according to grants, is the publications, and he has about 150 publications in all the fields that really have to do with some role of genetics, particularly in the cardiovascular system. So a very accomplished individual, and we all feel very proud that you visited us today, and certainly the fellows in the interaction with you this afternoon. And I'm very pleased that you're going to be surveilled on an evolving field by Dr. Roger Hajar and all the great experts in the podium, and congratulations for your visiting professorship. I'm going to give you this. So I'm going to give you this check, this big envelope, small check.
Thank you very much indeed. It really is an honor and a pleasure to be here. Thank you very much.
Thank you. This is the faculty and the fellows. Yeah. Just saying that you are a great guy.
Thank you.
Okay. So. Yeah, let's put the fellows. Okay. Maybe. There we go. Thank you.
Thank you.
Okay, good. So now is so many experts here. So Roger, they're going to run this, John and Bruce. Well, two Bruces here. Bruce Darrell, Bruce Gelb. Amy's here. Jason is here. And Mark Miller, that today was promoted to the position of professor. So that was cheap, though. It was very easy. Is that right? Okay. Okay.
All right, so exciting topic and panel. So what we're going to do this afternoon is, instead of just focusing on atrial fibrillation, we will talk about atrial fibrillation and the burden of atrial fibrillation. But what we're going to do is really try to open it up to a little bit more CVD genomics as opposed to just atrial fibrillation. And we'll use atrial fibrillation as an example. So again, I'd like to extend our thank you for Callum to come to the Controversies. Callum and I shared a suite of offices many years ago, and really there was no one who worked harder than Callum that I know at the CVRC back in Boston. His light was always on. I don't know if you were in there or not, but your light was always on. Anyways, but again, thank you for coming, Callum. So we heard from the wonderful talk at noon today from Callum about some of the sort of directions that genomics has taken. And let's start out with the first question in terms of GWAS. And I'd like to ask Jason, who's obviously a clinician scientist who deals on multiple fronts, of his experience first with GWAS, and specifically how systems biology has kind of informed, I don't want to say the failures of GWAS, but really kind of the disappointment of, after spending so much money on these huge studies, where GWAS has taken us and where it will take us. Jason?
Thanks, Roger. And thanks also, Dr. Mcrae, for coming. It's a great question. I think sets the stage, and it's a vast question. We could easily spend the day talking about it. But just to recapitulate what GWAS is, GWAS has been the sort of backdrop to the last 15 or so years of genetic discoveries, which is basically taking what we call complex disorders like coronary artery disease, atrial fibrillation, obesity, hypertension, these types of complex disorders we're all very familiar with, and looking at interrogating DNA in cases versus controls to see what underlying DNA signal there can be to account for the burden of disease. And these have become increasingly larger and increasingly complex And current GWAS studies of coronary artery disease are upwards of over 200,000 people in these studies. So they're vast studies. And somewhat disappointingly, as Roger said, they've only accounted for in the order of about 15% of the heritability of these complex disorders. And that's due to quite a number of different considerations that we're only just beginning to come to terms with. So the vast burden of heritability has been unaccounted for by these massive GWAS studies. And one of the appreciations of GWAS is that it only really gives us the signal in DNA, which is obviously what we're born with, that we then interrogate to try and understand what the causal basis of disease is. GWAS doesn't take into account, for example, with coronary artery disease, the contribution of smoking, of the interaction of diabetes with your genetics, and so on and so forth. All the so-called environmental overlay signals that go into causing atherosclerosis are essentially missed by GWAS. So systems biology has come about as a way to capture that environmental signal, and that can be then intersected with GWAS signals and GWAS studies to try and come up with a more comprehensive understanding of the basis of disease. And I think a lot of us at this table are engaged in these systems biology studies of one form or another to try and move the field forward. And I would say that I think GWAS has been what's come over the last 10 years, and more or less we're hitting the wall with GWAS in many aspects, as the studies can only get so bigger to understand genes that are of lesser and lesser importance, versus now we're going forward with systems, which I think is really taking us to the next stage, where we're integrating these environmental signals with genetics to try and come up with a better, more holistic understanding of the genetic and genomic basis of disease.
Great, thank you very much. So, Callum, do you want to add anything to this sort of overview of GWAS and where it's taken us? And
Sure. First of all, I'd like to obviously thank Dr. Fuster, Dr. Goldman, and everybody on the panel for the opportunity to be here. It really is a distinct honor and privilege to participate both in morning rounds and then again in this evening Controversies. It's really a fantastic opportunity to come to such an incredible place. I think AF is actually a good place to start for GWAS. So if you think about it, the way I always look at genetics is you have to go back to the patient. And I actually chose atrial fibrillation as a place to think about human genetics for one reason, and one reason only. It's a paroxysmal phenotype. So it's the ultimate complex trait. It's one that you don't even have 90% of the time or 99% of the time. So it lays the groundwork for how difficult it will be to overcome some of the issues in genetics. The other, I think, important piece of this is that it means that a lot of the people that we think are not affected by the trait actually have the trait, it just hasn't manifest. And that's a key part of almost everything that you think about in genetics. There are two other things that I think help put GWAS in context is when you're thinking about it in general for complicated disorders. When you actually look at atrial fibrillation, you tend to find it in families with cardiomyopathy. So the trait itself doesn't breed true as a single entity, although it does tend to occur in settings where it is associated with similar manifestations, but it's often very tightly associated with cardiomyopathy. And in fact, the major Mendelian loci, the large family studies of AF, have shown that every one of the AF families maps to a locus that has also been shown to be important for dilated non-ischemic cardiomyopathy. So you then take that context, i.e. something where the penetrance is very low, there's a very high false negative rate, which we know already just from general clinical practice, and we know already that it's actually associated with an atrial myopathy, which we're not measuring. We're waiting for it to manifest itself. And you can see how genome-wide association studies would be remarkably tough. So you can find lots of signals, and in fact, the second largest signal in all of genome-wide association is in atrial fibrillation. That's the PIDX2 locus. It has about a 3x to 4x effect on your risk of atrial flutter and atrial fibrillation. Yet even in that population in which it was described, it still only explains about 10% of the heritable variants. So the core problem, I think, with GWAS is that the traits themselves are aggregates of lots of different disorders, that they are poorly penetrant, and therefore there's a high false negative rate in the controls. And then the third thing is that we make the assumption that they are really powerfully relating what happens in common effect variants with what happens in rare effect variants. And I would argue that that works very well for quantitative traits like lipids or biomarkers of other settings. But for yes-no trait, for binary traits, it's not a powerful tool because you're putting genetics at a disadvantage. You're lumping things together that you know already have discrete etiologies, which is sort of the first thing that every geneticist is taught to avoid. And so that's really at the core of what I believe is the problem with GWAS is we've spent a lot of money and a lot of energy finding signals that are so small that we will never be able to identify any causal mechanism in the traits that are binary. I think they might be more useful in quantitative traits. But AF is really a great paradigm in which to understand what the limits are. And it's usually a function of the fact that we're not measuring the thing that we're truly interested in, which is the underlying diathesis to the end disease, which in many instances for AF ends up being stroke and heart failure and not AF itself. So,
thank you, Callum. So, Johan, you've had some success being involved in multiple GWAS studies, atherosclerosis, cardiometabolic syndrome more recently, and clearly you've fished out some interesting genes. So why don't we just talk a little bit about that?
Yeah, thank you for inviting me to the panel. Yes, so we find that although the genetic signal found by GWAS are independent, I think about 90 independent loci for coronary artery disease, we find, as I think alluded here to already, that if you look at the mechanism, and we are interested in looking at these gene networks, they actually merge into the same network downstream of the genetic signaling. So for coronary artery disease, a large majority of these loci have common genes that they regulate both on the cis and trans level. Also, I think that we have found that some of these loci are not at all shared between different, not specific to specific tissues. For instance, the PCSK9 loci, we find actually the risk mediated in that loci is not going through PCSK9 regulation in the liver, but the visceral fat. And in general, from the functional studies we do, the role of fat tissue emerge as really important as a key regulator of not coronary artery disease only, but cardiometabolic traits. So what we are seeing is that although these genetic signaling might be specific or independent in themselves, they share genes across tissue. There are many mechanisms underlying these loci. And also there are shared genes across different cardiometabolic disease, including lipid disorders and similar. So that's our finding. So I think it's a long journey, and as Jason alluded to, I think the GWAS data sets are great data sets, but I think as up to date, the way we have chosen to analyze those data sets, which are really looking at the tip of the iceberg due to vast multiple testing problems. So the real challenge is how do we find more of this missing heritability in these GWAS data, and how can we extract the true variants that contribute, although they may not be at the genome-wide significance level. So something like that, I think.
That's great. Thank you. I think that's an appropriate introduction to GWAS and what's happening, at least at some degree, at Mount Sinai. So one of the most important things as we put these data together is really phenotyping, and whether it's clinical phenotyping from physical exam or imaging or lab test. So there have been large studies looking at how to best phenotype and how to correlate this data with genetic information. So, Bruce Gelb, tell us a little bit about BioMe.
Sure, be happy to do it. So hopefully people are aware that we have here at Sinai the Bronfman Institute for Personalized Medicine, and it was really Erwin Bottinger who had the foresight a number of years ago to get Mount Sinai started on what has now become a very important enterprise, not just here, but in many places, which is biobanking of individuals. And one of the very smart decisions that was made for that, and at the time put us in a different place than a lot of other institutions, was that as we enrolled individuals, we didn't do it, shall we say, the easy way, which is just a check-off, like you were in the hospital, you gave blood, and if they didn't use it and you didn't say you didn't want us to use it, we went ahead. Instead, they actively consented everyone and said, can we look at your medical record from here till eternity? Yes, and most everybody allows that. And if we need to, do you mind if we call you back? And again, almost everybody has said yes to that. And so then, over the ensuing years, something on the order of 40,000, in excess of 40,000 individuals have been collected. There are bigger collections. Vanderbilt's certainly bigger, but we have ramped up, and now we're shooting towards a goal well in excess of 100,000 individual. Sadly for me, almost none of them are children. They're almost all adult patients. But that has really positioned us, because of the wealth of information we have in our medical record, to combine it with the genetics. So a particularly interesting moment has arrived at Mount Sinai within the last month or two. So through a partnership with Regeneron, in excess of 30,000 of those 40,000 people have had whole exome sequencing. Now, it doesn't give us the genome information, but it does give us every bit of coding variation, which at the present time remains what's most easily understood. And that allows us, and Amy and myself and others, we're working with Ron Doe and others, are for the first time able to ask in a sort of large population-based way questions driven by genetics first, things that we haven't known. If you think about how genetics has worked, for the most part, it has been taking people who have a disorder and studying their genetics and making conclusions from that. But we haven't always had the opportunity to run the experiment in the opposite direction and say, I don't know what, to pick your favorite gene, if we take everybody who has variation in that gene, particularly ones that we think are damaging, how many of them actually have the cardiovascular problem we think they ought to have? And if they don't, why not? And it also gets the idea of how many people are we diagnosing correctly for the Mendelian stuff. But it enables a wide range of things, certainly for the simple genetic disorders, but also for the complex traits. And so the kind of findings that have been alluded to here through GWAS can also be interrogated in that way. And the last thing I'll say is that, and anybody who works here knows that we are blessed by having an incredibly diverse population whom we serve, and that means that the genetic information that we're getting will be quite different than they would get at other places. The group that's in front of us in terms of this similar sequencing is the Geisinger Clinic in rural Pennsylvania, and we can understand that their population makeup is quite a bit different than ours. So.
Great. So the interrogation is at the level of the genetic information, and then you can correlate it back to...
Yeah, you can run it in either direction. You could instead say, I'm interested in, name the cardiovascular phenotype, pull from the medical record information, most easily from ICD codes, but there are other ways with natural language processing, pull out everybody who has that phenotype, and then ask the question, what do they share genetically? Conversely, where we know genes, we've had this problem around cardiomyopathies, for instance. You can take all the cardiomyopathy genes and say, okay, if I take everybody who appears to have a change that really ought to cause cardiomyopathy, how many of them have heart problems? Is it only cardiomyopathy, or are we seeing an excess of other traits, cardiovascular traits or other traits in these people? You can ask many, many questions. It's a very rich opportunity that we have right now.
Thanks. So Bruce Darrow, so in terms of EMR, that's going to be obviously very important to have quality control of the phenotype, the clinical input that's there. So how is that database connected to the genomic database?
It's actually a real problem. You start with the premise that nobody ever comes to me and says, you know, I'd like to do more clicks in my EMR. Then just the complexity of what are the phenotypes of atrial fibrillation. Is the person who had atrial fibrillation for 5 minutes on post-op day 4 from an off-pump cabbage the same as somebody who has paroxysmal AFib over the course of, several years de novo. And, if I break my hip and somebody fixes it, I no longer have a broken hip. If I have AFib and somebody ablates it, how am I going to document that? Do I still have AFib? Do I have former AFib? Am I cured of AFib? So there's a lot of complexity behind the phenotype of AFib that not only is difficult to fully express the proper nuance of it in something as unforgiving and black and white as an electronic medical record, but it would be hard to get everybody in this room to agree on simple answers to very common questions about how to list the phenotype. So it's, the easy way, as Dr. Gelb said here, is to just pull off of ICD-9 or ICD-10 codes and say, well, you had AFib at some point. But, and then we can use that and we can look at a biobank, we can look at a genetic repository, but the quality, the heterogeneity of the information behind that single diagnosis of AFib is more complicated than I would wish it.
Thank you. No, I think that's a very important point. As you mentioned, do you have AFib after it's fixed, after it's controlled? And that applies for multiple phenotypes within the cardiovascular realm. So, I mean, Callum, if,
can I ask one quick question?
Yeah, sure.
Just a second. Do you have standardized documentation across the organization? Do you have a fixed documentation strategy? No.
We have a strategy which is not complete, but about 80% of the enterprise is now on a common platform.
But the way in which individual providers enter data is not standardized.
Have you tried working with 6,000 doctors?
Oh, yeah.
They have their own ideas about that.
No, I was just wondering if you had done something that nobody else has ever been able to.
Sadly, I can't take credit for that.
Okay, so Callum, I mean, we obviously know the environmental factors, nutrition, all those things are important and have or seem to have some effects in terms of outcome, even with a genetic background that could be extreme. So I think we have a utopian dream that our supermarkets are going to be the health insurers of the future. And basically, even what you buy as food is going to be documented and going to put it into this database where, if you have a specific type of genetic background or polymorphism, you should not be buying this, or you should be buying something else. So where are we with this sort of big data information and manipulation that is going to get us to that stage?
So I think we're quite a bit away from it, but perhaps not as far away as we think we are. So I think it actually gets to Bruce's last point, which is, I think one of the problems with almost all of healthcare is we've added this subjective element to almost everything that we do, and that's us. And there are areas where subjectivity is really important, but there are areas such as big data where it completely kills everything you're trying to do. And so I always make the analogy that the medical record is like, trying to predict things from the medical record is like trying to predict what movies you want to watch through Netflix, except for the fact that you're not even a Netflix subscriber. That we're sort of true and vaguely related to reality. And the same thing, I think, applies in spades in nutrition, where the gold standard for essentially 45, 50 years has been the food frequency questionnaire, which even the nutritionists have shown is almost irrelevant. It doesn't even, your food frequency questionnaire on day one doesn't predict anything about day 2, 3, 4, or 5. There are strategies that I think in most of the rest of our lives where we may collect information that is not necessarily perfectly representative, but is still much more information rich than anything that we currently do. So for example, if you think about a supermarket, obviously it's tough to know who's going to eat exactly the food that you buy in a supermarket. So a supermarket's probably not going to be able to identify that all of that six-pack of beer was actually going to be drunk by me, although they can probably make a reasonable inference if they've seen my wife. On the other hand, what they can do is look at things over time and make predictions from data that may or may not be perfect reflections of what we imagine. And I think that's already happening. You know already, you only have to log into one website once or visit one store once to have your entire internet experience for the next 3 or 4 weeks dominated by adverts for that particular technology. And so these types of strategies are already being deployed in large supermarket chains around the world to predict what you might want to buy next to help them with their supply chain. So it's only a small degree of separation further to actually be able to work out not necessarily exactly what you're eating, but roughly what you're eating, at least much more reproducibly than what is happening in traditional food frequency questionnaires or traditional epidemiology. But I think the problems are exactly illustrated by this, that really rigorously and quantitatively robust data that are imperfectly related to what we really want are actually much better than semi-subjective nonsense that we believe is perfectly related to what we want. And that's ultimately at the core of almost all of the EHR data. We actually firmly believe that the current EHR is so vaguely related to reality that you have to actually restart from scratch, building objective data. We did an AI project where we tried to identify heart failure patients from partners' EHR. 196,000 people in a particular domain were labeled as having heart failure. We could find objective documentation of heart failure in 5,600 of those 189,000 individuals. So that's the problem, is that if you think about it even in terms of what we do as docs, when I was a resident, when you and I were, well, you were, I think, a year ahead of me, but when I was a fellow at MGH, maybe a little bit before that, 2% of all heart failure was preserved ejection fraction. Now it's 50%. All that really changed is the billing for heart failure. Nothing else has really changed. The diagnosis is not any more or less common than it used to be. It's just that you get paid much more for shortness of breath if you charge it as heart failure than you do if you call it something else. And so those types of diagnostic trends are what has swayed most of what happens in medicine. Everybody knows that you can't measure diastolic function using a load-dependent index like an echo, but it's now the standard of practice. I've never seen an echo with a load measured in my entire life, and yet it's now standard of care. So those are the types of sort of elisions that happen both in nutrition and in the EHR that make it almost impossible to use the current data sets for any form of real large analytic program. So we're going to have to start from scratch is basically the problem.
Okay, so yeah, well, that's a little depressing to hear. So I guess we just need real tissue and real data from actual biobanks. So Jason, back to you.
I mean, I would just say with BioMe, just to digress a little bit, it depends what trait you're looking for, and clinical traits are very challenging, but from BioMe, you can query things like cholesterol levels, which are robustly measured and recorded, and a couple of us, height, a couple of us have been involved with some BioMe projects where we actually looked at LDL, looked for rare variants that were associated with outliers of LDL, very high, very low, and we've had some modest success with that approach. So I think there is mileage in the BioMe, but it depends on what trait you look for. As far as tissue collection and moving forwards in that direction, I think there was some serendipity, and I like to believe I had some good planning involved as well, but I launched 2 clinical initiatives years ago, fibromuscular dysplasia, where we set about collecting tissue biopsies of skin and deriving fibroblasts to look at fibromuscular dysplasia. And also Catherine Michaelis, who just left, spearheaded a project where we were collecting surplus tissue samples from patients having various operations, and we ended up collecting the healthy aorta from transplant donors, the diseased aorta and the internal mammary artery, and we leveraged all of these tissues, fibroblasts with fibromuscular dysplasia, and these surgical samples to query atherosclerosis, and particularly mesenchymal stem cells in the context of atherosclerosis. And using various systems approaches have been quite successful and in drilling down on the cause of these two diseases. Fibromuscular dysplasia yet to be published, but Catherine Michaelis's paper comes out next week in one of the Cell Family journals. And that was an inspiration to me to really push ahead really with this tissue collection in the context of cardiovascular and cardiology. And with Dr. Fuster's support, your support, Roger, we've actually been successful in coming to the point after several years of planning of launching an adult cardiovascular tissue biobank and omics facility that we've actually found a manager and in the process of onboarding a manager for this in the next coming weeks. So going forward, you're going to be hopefully hearing a little bit more about our cardiovascular tissue biorepository, which has the full buy-in of David Adams, the cardiothoracic surgical department, Michael Marin, the vascular surgical department, Raja Flores from thoracic surgery. And we look to be building a tissue biorepository and some fresh tissue collection so that we can do fresh tissue processing to really try and leverage some of the other great strengths of Sinai, such as the genetics and genomics facility, to really push ahead in these systems approaches with cardiovascular diseases.
Great, thank you. So Callum, you know, in your talk there was one sentence that I really sort of appreciated, and it was genomics data require genomics or gene-associated therapies. And obviously that's one of my favorite topics. Now, Amy, how can we model some of these genetic information using IPS, for example, and that's when we can deploy in a humanoid type of fashion some of the genomic therapies and gene therapies and biological therapies?
Sure. So everyone has heard the term personalized medicine or precision medicine, and I think the enthusiasm is there, and we're slowly working to get the science to meet the enthusiasm. One of the exciting fields is in induced pluripotent stem cell research, where cells that are derived from a patient with a particular genetic background can be expanded in vitro and then used to model a disease. And that's been well described in a number of phenotypes. And in atrial fibrillation, I think the challenge has been over the last few years, you know, the protocols for differentiating a human cell, a terminally differentiated cell, making it more stem-like, and then redifferentiating it into a cardiomyocyte. That part has been more or less worked out. But atrial and ventricular cells are unique genetically and phenotypically, and we know now that they're derived from different mesoderm subtypes. And so that's been a challenge that only recently has been worked out. And now that we have that as a modeling system, we can look at specific effects of antiarrhythmic therapies on atrial cardiomyocytes in cells that are derived from individuals with different genetic backgrounds. So that empowers us to study patients who either maybe have the rare Mendelian form of familial atrial fibrillation, understand how different antiarrhythmic drugs work in those in patients with those mutations, and also in the different SNPs that have come from the GWAS studies. And also to then look in the ventricular cells to see the effects of these drugs in inducing early after depolarizations or some of the negative side effects that have pulled other drugs from the market or prevented them from going to trial. So this is the concept of a clinical trial in a dish. And I think it's an exciting way of preempting studies before they go into humans and sort of tailoring a study so that it has the highest likelihood of having a statistically significant result. And then, of course, there are other questions that can be asked about pharmacogenetics for anticoagulation, but I think that might be for a different discussion. Anna covered that in her paper.
Great. So Johan has a question.
Actually, I have a question, but I thought maybe an addition to the debate. So I also want to say a few words on the third task. You know, educating the population about what they can individually gain from gathering their own omic and genetic data. We're starting to see that. Back in Sweden, 2008, I started a 23andMe kind of company, and we actually had a discussion with the leadership of 23andMe and about what kind of test we should have, And then the geneticists came out and they were shouting that, oh, either the rare testing was too serious, it was too big of an effect, and the common disease, there was no effect, so how could you give this to the population? They can't decide. And I think there is something with us doctors and scientists that we are sort of offering genetics to the population, where actually the revolution is going to come when the individual realize how important it is for to sort of gain your biological capital and understand who you are, particularly early in life, and you want to gather this information in your own accounts, and then we will become more providers, and we will ask these people to participate in research and in health, and we will, more like an Amazon-driven kind of way, provide services to the patients and the population. And I think this is something that we are not doing so well, trying to educate people for them to understand what kind of insights they can get from gathering their own genetic information, and I think that's a big task ahead.
Well, most people seem to be interested about their ancestry and where they come from, but yeah, clearly that's, I mean, that's interesting, but not clinically relevant in certain cases. Bruce, you started the whole field of disease in a dish, so you have any addition to the precision medicine?
Sure. So, I mean, I thought Amy laid out very well the opportunity. I'd add a couple things. One, another major advance that's come in that area has been the ability to make tissues. For quite a while, we were left just looking at cells, just in flat culture, and we know that that's interesting, but nowhere near what a heart is like, really. And now we are getting to the point that people are making tissues, they're functional, and they can be done in parallel in numbers that would allow you to do things like look at drugs. So that's certainly coming along. On the flip side, we have not yet found the key that unlocks the door to mature cardiomyocytes. We're still looking at relatively immature cells. We maybe have moved from embryonic up to fetal, but we're not quite up to even a newborns yet. And I think for some diseases, if you think about AFib, I think that it's a little hubris to think that we're going to model what real AFib is that typically comes on multiple decades into life in cells that are more similar to something before birth. The second point around that is that we know that although cardiomyocytes are important, they're hardly the only cell type in the heart, and there are all sorts of interactions, and we're still working on getting that modeling right. I mean, you can pour different cell types in there, mixes, and people are, and my group's been looking at that a little bit, but it's not straightforward. And I guess, though, that's excitement, right? And for younger people, there are opportunities. There's so much more to advance. It's a new field in the scheme of things. But I think we have to balance between its potential, which is enormous, with not overselling what we're able to do today and the need to bring it along a little further.
Great, thank you. All right, Mark. So, atrial fibrillation, from a clinical point of view, what's a burden?
What's the burden?
Of atrial fibrillation.
In regards to?
General cardiovascular population.
Well, I mean, obviously, it depends on how you see, but it probably affects, 15, 20% of the patient population. And as regards to genetics, I think the points are well taken, which is that this is a disease much later in life, and atrial fibrillation itself is probably just a manifestation of an atrial myopathy more than its own disease entity itself. And the interesting, when someone had brought up about the idea of how do you define AFib, and you could probably break it down in 100 different patient populations. You have persistence with lots of left atrial scar and persistence with no atrial scar, and so those are different patient populations. And versus a paroxysmal who has vagal-induced AFib at the age of 30 versus a paroxysmal who has true AFib at the age of 30. So I think that from at least my basic understanding of the genetics of AFib is that I agree that within cardiomyopathies and familial AFib, there's probably some component to it, but I have to imagine that the rest of it is a disease of aging. I'm not saying there's not a genetic component to it. I just don't know what a 65-year-old with AFib is necessarily going to get from a genetic analysis, or if that will ever lead to a change in his or her particular therapy. But it would be interesting to be able to divide patients and understand why they're so different.
So from a treatment perspective, I mean, we see now this term atrial cardiomyopathy or atrial myopathy pop up a lot more. Fibrosis seems to play a much, more central role in atrial fibrillation. Does that help in any way? I mean, obviously from there you can go back and look specifically at some genetic component. Would that help in any way, regardless of the age of the patient, the type of treatment that you would deploy?
Well, I don't know about specific treatments, but it would be interesting to know whether or not patients select out based on their genetics for patients who are treatable for their AFib. As Bruce pointed out, we can ablate anyone for AFib, but clearly some patients are not curable. And so if genetics is playing a role in the patients who have a worse disease state, such as no different than if someone has a non-ischemic cardiomyopathy due to a genetic mutation, maybe there are patients who have AFib due to their genetics, which no longer allows them to be treated with the standard catheter ablation or antiarrhythmics. And to Amy's point, there has not been a new antiarrhythmic for AFib in many years. And part of that is to the point that, for a particular trial, if you have a few percentage points nowadays of, negative outcomes, the drug will never be approved. The example always being that if dofetilide were brought to the market or brought to the FDA today, it would never be approved. So getting these drugs analyzed before they ever get used in a patient would probably be more helpful in selecting them out, so that people would at least feel motivated to test further drugs. Right now, there's no motivation in the market for drug therapy, at least it relates to antiarrhythmics.
So Bruce, you think with the atrial cardiomyocytes being picked out in IPS-derived fashion, you'd get more information about the atrium or how newer drugs can affect these types of cells?
I mean, I think the potential is clearly there. I think that it has been generally surprising how good the modeling has been. Now, there is literature bias. We only publish the things that work. But for instance, in the cardiomyopathies, again, things that are presenting not super late in life, but well into life, decades, are being modeled reasonably well. Now, whether that will be true of atrial arrhythmias, I mean, my group is working on an atrial arrhythmia. It's not been an easy project, but I think the jury is very much out on that. As a simple example, you don't get T-tubules in the cells we're differentiating yet, and so you got to believe the physiology of these cells is somewhat different than the normal physiology. And so, I think we have to see, but the optimism is that so far, the iPS-derived cardiomyocytes have proved better at modeling than one would have thought, just a priori knowing what we know about, what they appear like from how far into differentiation they are.
Yeah, no, absolutely. I mean, I think also putting the endothelial cells in there and trying to understand anticoagulation in the setting of paroxysmal AF is going to be part of that modeling, and I think that's part of the excitement. So anything else you wanted to add from the atrial fibrillation point of view?
No, I think the whole idea about treating SCAP, which is one of the new concepts that is being paid more attention to, is going to be very interesting. There are 3 trials that are being run right now that are looking at giving therapy and taking away therapy by device-detected arrhythmias. So the jury's still out on that as well. So that's going to be one of the interesting ones.
That's great. Well, from the ventricular side, we know that less scar is better overall. So we'll see. And with your ablation, you're causing more scar, right? I mean, is that
minimal? But to Anna's point, actually, which is interesting, is you have to imagine that in 5, 10, 15 years from now, we're not going to be anticoagulating patients based on their history of AFib. We're going to be anticoagulating patients based on their risk profile for developing AFib or the same risk factors that coexist in AFib. And there's a few trials being done now in device-detected AFib in patients who simply have risk factors for AFib, and then seeing what happens if you necessarily first is how often are they actually having AFib. And the next question will be, in patients who have enough risk factors to develop AFib-related stroke, should we just be treating them in the absence of AFib for stroke? So I think hopefully in many years we'll be able to use that information.
So Callum, 10 years from now, we're sitting at the same panel. What do you think we would be saying about genomics and cardiovascular disease or atrial fibrillation specifically?
So that's a good question. I think you've heard most of the threads that will be relevant. I think my sense is that phenotyping will be much more rigorous. We'll be able to phenotype the substrate, for example, for AF. As Mark says, we'll probably be prophylactically treating with ablation or drugs or anticoagulation. There are already, for example, stable substrates that predict AF much better than AF detection, but they're just not commercially available. And they're only applied to very small subsets. So they're very well-known co-segregation between AF and some panic disorders that are genetically identified. They're actually cutaneous measurements that predict genetic risk score for AF stably throughout your life. You don't need to do recordings. They're actually better at predicting your risk of AF than the genetics are, to be honest. And those things, I think, will be broadly implemented. That having a stable quantitative phenotype, as both Jason and Johan have outlined, will help the genetics. We'll understand the discrete mechanisms, and then hopefully we'll be collecting the data passively and integrating it in the EHR with all of the other information, including the amount of beer that you're drinking, directly fed in from the pharmacy or the supermarket. And then as the baristas say, you'll be able, to identify who those folks are from an aggregate of data that people have seeded to you for that purpose. I mean, if you think about how we use, everybody's freaked out about giving their healthcare data, but we give people their social security and our credit card data every day because we're getting something valuable in return. If you're getting that in return from day one, you're going to be willing to give people almost anything you want. And then I think the final thing I would say is if you think about a genome, the thing that's really made genetics successful and genomics successful is it's the only bounded data set in all of medicine, in all of biology. It's the only one where you know the start and you know the finish. Everything else is almost infinite. There are an infinite number of environmental factors. There are an infinite number of cellular connections. There are an infinite number of so many other different elements in care. There are only a very fixed number of genes, and we know exactly, and intergenetic material. And it allows us, therefore, to organize everything else. And if nothing else, I think the genome will end up being a powerful organizing principle for the EHR of the future. That you'll go into it knowing, you won't know exactly what's going to happen, but you'll know exactly what the baseline was. And that will be actually a very useful framework for understanding things. And I will tell you that I think everybody in the world will have their genome sequenced before we know what to do with a single one of them. So.
Great. So, I mean, as a disclaimer, we didn't really talk about epigenetics, RNA sequencing, and all the tools of long non-coding RNA, microRNAs, and I think that just becomes a little bit too much. But I'd like to just open for everyone to ask a few questions for our panel and for Callum, if they have any. Dr. Fuster?
Looking at the cardiovascular field in general, there's no question that personalized medicine is a very attractive movement. And I think we are really behind in terms, compared with cancer and so forth. But I'm very, very concerned about the fact that we are missing the roots of the problem. That is, for example, in the United States, I just was reading an article yesterday about personalized medicine in terms of genetics and hypertension. When 50% of the people who have hypertension, they don't know they have it. The same is, I was reading what is going on with the use of drugs that we need to use in people with coronary artery disease, and about 25% to 30% of people are not using it. And even in trials like our Freedom trial, 50% are not using the medication we're supposed to use. The only reason I'm saying this is that I'm all excited about personalized medicine and so forth, but I think there is a problem in our society, and it is not acknowledging a huge issue, that unless we talk about it, it's completely the opposite of personalized medicine. It's just what you might do, and so forth. It's just a comment.
I think that's actually a very apposite comment. I think one of the problems with modern medicine is most of the variation we measure is actually variation in the input. It's how well treated the patients are. There's such heterogeneity in what people actually receive, that a lot of the downstream measurements in the EHR are a function of how haphazardly the interventions were made. And I think your point is very well taken, to the extent where I actually think until we devolve the guideline level care to a system where it actually just gets implemented systematically, most of what we measure will not really be anything more than understanding the artisanal nature by which doctors deploy different therapies. We found exactly the same. We found Blue Cross Blue Shield patients in Massachusetts, at least 30%, and maybe as high as 40%, had never had a second statin prescription written after discharge. So the only one they'd ever, after an MI or a stroke, the only thing they'd ever had was that initial statin prescription. And then we wonder why the recurrence rates are so high. In the partner system, if you weren't in a advanced heart failure clinic, if you weren't in a transplant clinic, you had only a 20% chance of being on more than 2 of the 5 drug classes recommended. And of those people, only 20% were at the optimal doses of any of the drugs. So unless we start to use the things that we know already work, all we're doing is measuring how poorly implemented things are in the residual population.
But I think Dr. Fuster's point also is that even when you prescribe these medications, they're not even taken by that.
I agree, but I think some of that is the way that we deliver it. If we're not even willing to write a second prescription, if the system is so haphazard, then all that tells the patients is we don't really have a vested interest in making sure that this drug really gets taken. So we need to build strategies, as people are doing, to measure compliance, to measure the responses to drugs in real time. And then I think, as Dr. Fuster was alluding to, we need to educate everybody that this is why we're doing it. We're doing it because we have evidence that this actually works. And until we do what we know works, it's very tough to get people to test or participate and be engaged in testing things like genomics that we're not really sure of how they're going to apply.
Marty?
Excellent panel, very good job, Roger. So one of the potential problems if we're led by the genome and genetic data is stigmatizing patients. And since we know that there's such a big gap between the genotype and the actual expression of the disease, we end up over-treating patients or stigmatizing patients with significant disease entities, which will never really become a reality.
Well, first of all, on this very day, the New York State legislature managed to pass a really improved law around anti-discrimination by insurance companies based on genetic testing. So, I mean, I think at the end of the day, what's required is that culturally, particularly from companies, insurance companies, but also physicians, that there's a full understanding of the difference between genotype and phenotype. That to carry a gene that makes somebody susceptible to a particular problem is not the same as having that problem. And particularly as we're identifying variation of varying degrees of strength and penetrance and so on, it's really important that we get that right because the risks are enormous. There's no doubt. And if we're labeling everybody who's carrying a variation that might make them susceptible to certain electrical, the channelopathies, take Brugada where the penetrance is so very low. I mean, to create havoc around that is just not doing our job. So I think it's going to be on us. We are in medical schools. We are educating the next generation, and we have to make sure that physicians broadly understand this. And we also have to communicate it and be good partners to, insurance companies when they want to have conversations and to the public at large. That's my view. Colin, what do you think?
So I agree with everything you said, Bruce. I would add a couple of things. One, I think we, as a community, managed information less well than some other communities. So, for example, if you look at the life insurance industry, which is the only one that can really be impacted by genetics, because of GINA, you can't be discriminated against in healthcare, although you could be discriminated, as you point out, Martin, based on doctors over-treating or under-treating genetics. But most insurance companies have already factored in your genetics without measuring it, based on your family history. So, and in fact, it's a good illustration of big data. So the life insurance company factored in tobacco into life insurance premiums between World War I and World War II. It took Dahl and Peto until 1959 to demonstrate the relationship epidemiologically, but it was already fully factored into life insurance in about 1913 or 14. And so I think that just tells you that some of this is how you amortize risk across large populations. Some of it is how you personalize it. I think we as a community are going to have to really make sure we're testing things as rigorously as we can. And I think that probably the one other thing I'll say, because it relates to big data, if you look at the average web page, because the way the web pages are set up is systematic, Google is testing 5 randomized controlled trials every time you open a web page. And if you think about the fact that that's built into their business, we have no means of testing anything in our business without setting up a completely separate infrastructure, rolling out at great cost contractual research organizations, measuring something, and then winding the whole thing up. If we actually had a system that measured things, then we wouldn't just be studying the stuff that drug companies pay us to study or the NIH or other foundations pay us to study. We'd be studying everything in real time, and we'd have answers much more quickly. And so I think that's really at the core of what we need to understand genomics and everything else, is a system for collecting data and implementing care that's truly a system. It doesn't depend on who you see, what day of the week you see them. You get the same treatment. I mean, I spent a day with a billionaire by chance about 6 or 8 weeks ago, and he basically said, the thing you guys need to fix is the fact that I can take the same data, the same person, to 3 of the world's experts on the same day and get 3 completely different answers. He said that for a simple problem for which there are actually hard data just embarrasses you as a profession. So that's not to minimize the role of the investigator or the investigative physician or the true professional. But I think what we've done is we've held on to things that are systematizable and therefore made ourselves unable, we don't have the bandwidth to deal with the problems we actually need truly informed professionals helping you interpret ambiguity. We spend most of our time doing stuff that literally should have been devolved to another system many, many years ago.
Great. Well, on that note, I think we can close. Thank you very much for the panel and for everyone coming here. Back to you.